A very warm welcome to everybody, wherever you are in the world. And thank you very much for joining us. My name is Jeremy Farrar. I'm honoured to be a fellow of the Royal Society, and I'm here to introduce what is a wonderful prize. The Royal Society Africa Prize is one of a number of the Society's awards and medals and prize lectures which recognise excellence in science and technology around and across the world and celebrate outstanding scientific achievements and, of course, outstanding individuals. Nominations for the Society medals are open on the 30th November, and, of course, details are all available on the Royal Society website. But it's a huge honour to be able to introduce tonight the Royal Society Africa Prize, which recognises research scientists based in Africa who are making an innovative contribution to the sciences. And I have a lot of great things I do each year, even during this COVID time, but this I have to say is one of the highlights. It is to introduce Professor Warimwe, who will receive the medal, and we're about to meet in person. He will receive also a grant from the Royal Society of, I think, £15,000 to the research project and a personal award of £2,000. Tonight's talk, Fighting Diseases with Cross-Species Vaccination, is given by Professor George Warimwe, winner of the Royal Society Africa Prize for 2021, and could not be coming at a more important time in global health. Professor Warimwe is an associate professor at the Nuffield Department of Medicine of Oxford, based in Kenya. He focuses on viral infections that are transmitted between humans and animals in Africa, and critically vaccine development for their control. Whilst many of these viruses were first discovered in Africa, little is known regarding their distribution, their associated disease burden, and their viral genetic diversity in the continent. And amongst many projects, George's research team are at the forefront of also addressing the global shortage of yellow fever vaccination supply, work which will have a major impact on the number of doses that be based and the global stockpile of yellow fever vaccination, which even as we all focus to some degree on COVID, we should remind ourselves that the other critical emerging infections and re-emerging infections have not gone away, of which yellow fever is one, of which there is a chronic shortage of vaccination. But his recent work as well on COVID-19 serology in Kenya has informed decisions of a national, regional, and global response, and remains one of the most comprehensive seroepidemiological studies of COVID-19 on the continent of Africa, indeed around the world. Remind everybody, the audience, that this is a live Q&A will take place after George's talk, and we'll try and take as many questions that you raise in the Slido and then through the chat bar here as we can after George's talk. So with that, pass away to George. George, it's a huge honour to introduce you, and a great, great pleasure. And I hand over to George now for the Royal Society Africa Prize lecture.
Thank you very much, Jeremy, for the kind introduction. And I'm really grateful to the Royal Society for this prestigious award of the Royal Society Africa Prize. It is a real honor and testament to all the people that I work with in Africa and beyond on advancing vaccine development for diseases that are a priority in the continent. I'm going to be talking about animal health. This is an interest for me because I'm a veterinary surgeon, but it's talking about animal health from the perspective of human health. That will make sense as I progress through the talk. So animal bring us pure joy, as can be seen through the face of this Borana girl holding her pet goat. They are important for us because of nutrition, meat, milk, and so forth. They're also important for us because of they provide financial security. And this might be from the sale of animal produce or the animals themselves. And some of this financial security is important for other sectors like the education. I'm standing here because I was brought up by my grandparents who kept animals, and they sold the our prized bull for me to be able to attend university. So there's a real connection between animal well-being, animal health, and human health, and even human prosperity. The other thing we share with animals are infectious diseases. In fact, 60% of all known human infections are acquired from animals. That's a really big sort of statement about the importance of animals. So controlling disease in animals is actually really important, not just for the animal welfare and food security, but it's also important for public health. I will talk about a virus called Rift Valley fever virus. This is a priority virus because it causes epidemics of diseases in Africa, and it has the potential to cause a public health emergency. For this reason, the African Union and the World Health Organization, WHO, have prioritized Rift Valley fever, or RVF, for urgent development of vaccines and therapeutics, because we don't have any of those for humans. The virus is an RNA virus. We now know about RNA viruses, such as the SARS-CoV-2 virus, but this falls in the family Phenylviridae, and it was first described in Kenya in 1931, following a massive outbreak of abortion and really severe illness in livestock that was also associated with disease in humans, and we'll see about that in the next slide. So this slide just illustrates the what happens with Rift Valley fever and how it's transmitted and the disease that occurs. On the top left, you have an illustration of heavy rain and flooding. Now, outbreaks of Rift Valley fever coincide with very heavy rainfall and flooding. And this is because mosquitoes are involved in the virus transmission. And so when there's very heavy rain and flooding, the numbers of mosquitoes increase. And this then provide a vehicle for the virus to be transmitted between animals and humans. What you're seeing there is just an Aedes, an illustration of an Aedes mosquito. It's just one type of mosquitoes, but actually we know that there are many types of mosquitoes that might be involved in the transmission of Rift Valley fever. So when there are outbreaks, the mosquito will transmit the virus between animals, and these animals, once they're infected, they'll have very high viral loads of Rift Valley fever virus. And so there's lots of virus circulating in the animal population. And these mosquitoes can then transmit the virus to humans. But actually, the single most important risk factor for disease in humans is handling infected tissues and fluids from affected animals. So because when RVF affects animals, you get very high levels of abortion among pregnant animals, and the very young animals, almost all of them will die. So it's a really nasty disease for livestock keepers. There is evidence of virus transmission in the wildlife, although the impact of the virus on wildlife is not fully understood. When the disease goes into humans, the symptoms you would see are very non-specific, muscle pain, sometimes, what is described like flu-like symptoms. But you do get severe disease as well. And this has been associated with very high levels of mortality, especially when you have hemorrhagic manifestations, which is one of the sort of symptoms of Rift Valley fever, severe Rift Valley fever. Why is RVF most common? This figure basically illustrates where cases of RVF have been reported in humans and livestock, on the left and to the right, respectively. And the more yellow the color, the more the number of distinct outbreaks of Rift Valley fever have been reported. What's very clear is that this is a disease that's specific for Africa and the Arabian Peninsula. And the distribution, as you will see in livestock, is a lot more widespread than it is for humans. Now, we talked about livestock being a big risk factor for human disease. So what's evident here is there might be more sort of underreporting in human surveillance compared to livestock surveillance. The other thing that you'll appreciate looking at the scale is that Kenya and South Africa are among the most affected countries, although we can already see, at least from the livestock data, that Western Africa countries are also prone to severe cases of RVF. I mentioned that we do not have any specific therapeutics for Rift Valley fever. We do have licensed vaccines for veterinary species, and this tend to be inactivated or live whole virus vaccines. So you use the whole actual natural RVF virus or some sort of modification of it to develop a vaccine. And these have been licensed. But there are still some concerns with some of these vaccines. Some can still cause disease. For instance, some cannot be used during pregnancy, or they need multiple vaccinations to elicit a strong immune response. They need very high containment to produce them, because they can, you can inadvertently initiate an outbreak because you're using the whole RVF virus. And some are not DIVA compatible, meaning that you cannot differentiate infected from vaccinated animals, so DIVA, because the antibody profile in targeting the whole virus is similar to that elicited by these vaccines. And there are no licensed vaccines for human use. However, there's reason for optimism in terms of developing a vaccine that can be used in humans. So what you're seeing here is a very old experiment from the 1930s, where you have serum obtained from TWN, case 1. I hope you can see that on the sort of second column of the table. And this serum was taken from an individual who had RVF, passively transferred into mice, group A2757, and the interpretation following a period of follow-up, after exposure of these mice to RVF virus, is that they were protected. None of the mice succumbed to infection. Whereas in another individual, SFK on this table, when the serum was transferred into mice and those mice exposed to RVF virus, the mice died on the second day, so indicating no protection. What this data indicates is that a humoral response, an antibody response, is sufficient to confer protection that is acquired following, recovery from RVF. And so what I've laid down there are three key observations that support this notion. The first being that you can passively transfer immunity via serum from immune humans to non-immune animals, as illustrated on that table. The other fact is that neutralizing antibodies that can neutralize virus from infecting cells, correlate with protection, and these have been observed in various animal studies. And the third thing is that when you recover from infection, you're essentially protected for life, because these neutralizing antibodies are really long-lived. They've been detected up to 25 years later following the original exposure, and having no period of exposure in between. So now that we know what we should target in developing an RVF vaccine, i.e. a neutralizing antibody response, and we know that this neutralizing antibody response actually targets the surface of the virus, so the GN and GC viral glycoproteins, we could then exploit this knowledge and basically transfer the coding information of the viral envelope glycoproteins into the ChAdOx1 viral vector platform to make our vaccine. And the original plan, from the outset, was to develop a vaccine that elicits very high levels of neutralizing antibody, not just in humans, but also in the multiple target species affected by the vaccine. So why did we use ChAdOx1? Because it's safe in humans. We already had very, very good safety information in humans up to that point. And now, of course, the safety database is upwards of 1 billion because of its use in the Oxford AstraZeneca COVID-19 vaccine. It can be manufactured to very high scale, so meaning that you can produce lots of vaccine and the process is already developed. The vaccine can also be thermostabilized. The vaccine platform can also be thermostabilized, which is important when you're deploying the vaccine in a situation where you don't have ample cold storage. So it would be able to survive outside cold storage for a period. So we have data supporting this. And of course, there's very low anti-vector immunity, meaning that unlike human adenoviruses, these chimpanzee adenoviruses, which ChAdOx1 is one of them, there's very little experience. Humans have been exposed to similar viruses to a less extent than they have for the human adenoviruses. So very low anti-vector immunity means that the vaccine performs well because you don't have that anti-vector immunity that limits vaccine performance. So what we then did, obviously, was to design and make this ChAdOx1 RVF vaccine, which on immunization infects the cells and expresses the coding information for the RVF viral glycoproteins, and this then stimulates the immune response. And the immune response we are interested in here is obviously the neutralizing antibody response. So this just is a schematic of that process that I've described. So we did the preclinical discovery that involves, identifying which antigen to put into the vaccine and how to manufacture it and so forth. And our plan then was to evaluate it initially in preclinical studies in mice, whether it performs as we predict it would. And then once we have that information, progress very quickly to livestock trials and human trials. And if all this works, then we would end up with a single vaccine that can be deployed in multiple species. So the first study, and the way I'm going to describe these studies is very much immunizing on day 0, allowing a period of up to sometimes 1 month. In the mouse study, we did it for 2 months. So allowing a period for the immune response to develop and then challenging the animals with RVF virus and then having a period of monitoring to look for disease, whether vaccinated animals develop disease or not. And you always have a control group that typically would be given placebo. So in the mouse study that we did, we got 100% efficacy against infection and disease. So the vaccine provided protection against RVF in mice. So this was very encouraging, but the true test really was to move into livestock. And we did this study in Kenya, where we looked at the 3 major species affected by Rift Valley fever, so sheep, goats, and cattle. Immunized them and followed them up and challenged them 28 days following vaccination, and then monitored development of disease. So it was really very encouraging to see very high levels of neutralizing antibody generated by the vaccine. That's what the graph represents. And the y-axis, it just shows that the neutralizing antibody titer. The dashed line represents the limit of detection of the assay. And of course, the 3 species are represented at the bottom of the graph. The colors are color-coded by the vaccine group. So we've got the ChAdOx1 in blue. Riftvax is the most widely used licensed veterinary product in Africa. And we included this as a positive control in our study. And the group in gray received placebo, so that we did not expect protection in those. So what you will see is that all the animals that mounted a neutralizing antibody response were protected, and all the ChAdOx1 RVF vaccinated animals mounted a neutralizing antibody response, and so this translated to 100% efficacy. There are a couple of animals that received the licensed product that did not seroconvert. This has been observed previously, and these were not protected, again, supporting the notion that if you develop neutralizing antibodies, then that is a clear correlate of protection. And the study was published in scientific reports as shown there. Next, we evaluated the safety and efficacy during pregnancy. Remember, I mentioned that this is an important, facet of the disease, whereby we don't have, vaccines that can be used during pregnancy, or at least most of them cannot. And we need to protect pregnant animals from, aborting their fetus. And this is what the study set out to do. We did it in sheep and goats, and this is a study that we did in the Netherlands with some of our collaborators. The study design was based on immunizing animals very early in pregnancy, when we know they are susceptible to any sort of perturbations in their physiology. And so a vaccination, if the vaccine is found to be safe very early in pregnancy, that's usually a very, sort of stringent test, at least had been used for other RVF vaccine programs. And then challenged 3 weeks later following vaccination. So all the animals did not show any sort of symptoms of being unwell, following vaccination. So the vaccine was safe. And we compared the performance of the vaccine here to a placebo, which we've called a mock vaccination here. The top graphs represent pregnant sheep, and the bottom represent pregnant goats. You've got the level of neutralizing antibodies shown there and the viral load. So this just indicates that very high levels of neutralizing antibodies were elicited by vaccination, and the ChAdOx1 vaccinated group did not develop any sort of viremia. So very high levels of protection. When it comes to the the fetuses, there were no signs of infection in all the 16 fetuses. So the 8 sheep had 16 fetuses in total. So there was a bit of twinning. And for the goats, we had 25 fetuses in total, and 23 of the 25 had no signs of infection. So this suggests that, again, the vaccine is very highly protective during pregnancy. It is safe. There is a need to look at the mechanisms of protection because of 2 things. So the first is that not all the fetuses were protected. For the goats, this might be, to do with the level of the number of fetuses in some of the dams. Some had up to 5. So which might, have an influence on the sort of the ability of the animal to mount an immune response. But also looking at the neutralizing antibody levels, there were higher levels elicited in goats compared to sheep. And we've observed this previously, and we still can't work out why that is the case. So, but it's some indication that there might be differences in the mechanism of protection between these 2 species. And also maybe the other big question is whether there's any influence on the stage of gestation on the immune response to vaccination, which these are questions that need to be addressed in future. So the final bit about the livestock development is this massive non-inferiority trials. It's done to very high level, high quality standards. So it's a veterinary GCP trial. And the aim of this is to support registration of the product in East Africa. And what it is a comparison of the performance of the vaccine, the ChAdOx1 vaccine, as compared to the licensed Riftvac vaccine that I mentioned earlier. But now in a study that is powered to look at the non-inferiority, basically we want to work out in the field, does the vaccine perform just as well as the Riftvac vaccine? And if this is the case, then we would have a very strong grounds to support sort of registration of this product based on it performing just as well as a licensed product, as well as the efficacy data that I've presented. And this is the results of this will be coming out later this year or early next year. So that's the veterinary development. And from this, we've actually generated a very large safety database of the vaccine in target livestock species. We've demonstrated that the ChAdOx1 platform can be used for indications for veterinary, veterinary use, so diseases that affect livestock. So, but how about humans? So, because RVF affects both humans and animals, so we can use the same platform, the same construct for development of the human vaccine. And this is what we set out to do. So in parallel to the veterinary development, which is really very advanced, we have already begun a phase 1 trial in humans. And the aim of this is really to add safety data specific to the ChAdOx1 RVF to the already large database of safety database of the ChAdOx1 platform from its use in the COVID-19 Oxford AstraZeneca vaccine. So this trial is ongoing in Oxford, and we have no safety concerns to date. The vaccine is performing well as expected based on the previous experience with this platform. And we'll be moving to Uganda for a phase 1b trial later this year. And the aim is to really just assess the safety of the product in humans and also the immunogenicity and to give us some confidence about further stages, further development, in humans. So in summary, we've demonstrated very high level of efficacy in livestock. And that the vaccine is safe and efficacious in pregnant animals, which is already addressing a major unmet need in the veterinary landscape. The safety and immunogenicity is expected to be as good based on previous studies or as current licensed vaccines, but the definitive data will be coming out later this year or early next year. And the vaccine is safe in humans, and these trials are ongoing. So where next then? So the focus of the next few years really will be to advance the human pipeline, and the 3 key areas we want to focus on or to identify what is the optimal vaccination regimen. So do you need a single dose? Do you need 2 doses? And what level of, how many virus particles do you need to elicit a high level of neutralizing antibody in humans? So, and then the second one will be on establishing a regulatory pathway towards vaccine licensure. So one of the most difficult things with RVF is that epidemics are sporadic. So it becomes very difficult to predict when you can do a vaccine efficacy study. And obviously, we cannot do a human challenge study because we don't have specific therapeutics, and also because there are debilitating sequelae that are associated with RVF, one of which is blindness. So you cannot deliberately infect humans for the purpose of evaluating vaccine efficacy, as is done for other diseases such as malaria, where you have effective treatments. So that regulatory pathway would be really important to nail down. And this might be through sort of working through the immune correlates, as I mentioned, or other strategies. And third, we need to work out a deployment strategy. So with a single vaccine that can be deployed in both humans and animals, in the face of an epidemic, do you deploy the same vaccine to both humans and animals at the same time? Or do you ensure high vaccination coverage in livestock and then, only vaccinate the high-risk groups? So these sorts of discussions need to be had and worked out to have a solid strategy. And this will involve, lots of different stakeholders, including the World Organization for Animal Health, the WHO, obviously, the African Vaccine Regulatory Forum, and obviously the actual vaccine end users, not just the people who would be vaccinated, who are targeted, such as the people working with animals, but also just the general public in terms of seeing the value of vaccines against epidemic diseases. I mean, this is a really important topic now and will continue to be in future. So I presented work moving from veterinary vaccine development, going to human vaccine development, and they seem like very different entities, but really our approach has always been to look at this as a single vaccine development pipeline, and this figure just illustrates the similarity between the veterinary and human vaccine pipeline. So we start with the target product profiling and immunogen design. So basically you're working out who do you need to provide protection to, what are the attributes of your vaccine, and those will be similar between the human and veterinary, for instance, for a disease like RVF. And you then move on to preclinical studies, as we did. And for the veterinary pipeline, you move very quickly to target species. And this already gives you lots of confidence with all the performance of the vaccine in this target species to allow informed decisions as you go onto the really expensive human development pipeline on towards licensure and deployment. And of course, there are cross-cutting regulatory and ethical affairs that need to be considered. So, but basically, the thrust of this is that any advance in one of the fields, whether it's veterinary or human, has direct benefits to the other. So for us, the large-scale sort of development or manufacture of the ChAdOx1 vaccine for use in COVID-19 is already of benefit for not just other ChAdOx1 vectored programs for human use, but also ChAdOx1 vectored vaccine programs for veterinary species, including the RVF program that I've just described. And of course, the information that we've gained through the sort of advancing the vaccine for the veterinary field has been informative in terms of what the regulatory requirements are in a country like Kenya. And these are cross-cutting issues again. And so we have learned quite a lot in terms of doing the work in my home country in Kenya, and how that then will inform a future evaluation of the vaccine in East Africa, and eventual deployment of the vaccine in this setting, which, as I showed earlier, is one of the most affected regions by RVF. And so this has involved quite a lot of work and lots of different collaborators, partners, and funders. And I would just like to give my thanks to all of them and welcome any questions. Thank you.
George, that was an amazing talk. I've heard you talk on a number of occasions, but actually not recently. And to hear the updated work was really amazing. So thank you very, very much indeed. I'm sorry we can't meet in person, but George, I bought you some flowers to celebrate the prize.
How kind of you, Jeremy.
So George, there's many questions, but if you don't mind, if I could just start up, I loved your story about being brought up with your grandparents, and I mean, what a great decision that was to make sure you could get to college. So your grandparents deserve great thanks and credit from all of us. A lot of questions though. Start with the epidemiology. What does really intrigue me is these, in this case, mosquito-borne again. But I spent much of my career working in Southeast Asia, never seen in Southeast Asia, indeed true of yellow fever as well. Is that a risk for the future, or do we understand why not? Why is Rift Valley still so concentrated geographically?
I think that's a good question. And I think one of the key things that actually maybe didn't come up from my talk is we do not have any evidence of human-to-human transmission. There's still a bottleneck in the transmission in that you need either livestock, that's one of the biggest risk factors, and mosquitoes. And when you're talking about mosquitoes, then you need the ecology to be suitable for that setting. So, but I think with the changing climate, I think there is always a risk that that you can get some emergence of these diseases in other settings. I mean, we've had things like blue tongue, vector-borne diseases spring up in Europe. African swine fever is now a big thing. So I think we cannot be complacent in, just thinking of these diseases as if they're just restricted to Africa. And of course, there's not much animal trade between, Kenya and I don't know, China, for instance, or other places. I mean, there is a lot of camel trade. The bulk of the dromedary camels actually come from Eastern Africa. And so that maybe explains the link between the disease burden in Arabian Peninsula and that being traced back to the continent. But we cannot be complacent.
Yeah, and also your links there between Asia and Africa are growing all the time, of course. And I think as with dengue as well, spread by similar mosquitoes, at least that may become a problem. I was intrigued that you said, I think you're right. You're saying 25 years later that you can still measure antibodies. Yeah, despite no intermediate infection. Is that right? That is a staggering.
It is. I mean, I think that's one of the things that really surprised me when I went into RVF vaccinology. Of course, I'd learned about it in veterinary school, and that the idea that an antibody response can last so long, it had never really occurred to me. But I mean, we know there are other diseases that do this. Yellow fever is a good example. But the original identification of the RVF outbreaks in Kenya. There were some of the investigators were accidentally infected, and then they moved back to England, I believe. And one individual 12 years, another 25 years later, still had neutralizing antibodies. And we've been able to replicate, well, at least in terms of look at the durability of that response in our setting here, where we saw that if you take this long-term longitudinal cohorts that we have at the KEMRI Welcome program, you can identify an individual seroconverting maybe 10 years before, from the time now. So it is definitely a long-lived response, and the hope is that you can elicit this response by, using vaccines, and that really is the main correlate of protection for RVF, because I haven't come across any study where an individual has an neutralizing antibody response and fails to be protected, following challenge. So I think it's one of those that's it's a low-hanging fruit, and I think the issue has just been the business case.
Right. And excuse my ignorance, George, but the variation in the virus, it's a single serotype. There are multiple serotypes. It mutates a lot. It recombines. Sorry, ignorant question.
So it's a single serotype. So as the antibody response cross protects against isolates from distant geographical settings, there is some variability in the M segment, which is where the viral targets of neutralizing antibody are, but that's around up to maybe 2 to 3% at the amino acid level. I think the thing that's a bit more risky for this family of viruses is reassortment, because they are segmented RNA genomes. It is possible that you get reassortants either from different wild isolates if they infect the same cell, if you have virus circulating from the very many species that are affected by this virus. So reassortants that perhaps maybe acquire more properties that are related maybe to disease severity or transmissibility is a possibility, and they have definitely been detected. But we still remain with a single serotype. Right. So we are very fortunate, and I think we have it might, thinking of COVID now, I think we have a very good opportunity here to target a disease where, vaccines seem to work, the target, It works very well, cross-protects against isolate from distant settings. So I think it's win-win before things get worse.
Fascinating. And just changing the subject a little bit onto the comments you made at the end there about vaccines in the veterinary sector. As you know, I'm not a veterinarian. How amenable, how easy is it, would it be, do you think, to persuade farmers, wherever they were, domestic or wild, to make use of a vaccine? Is that a huge challenge, or is there a demand for it?
I think there is a demand for it. I mean, the farmers, I mean, I talked about the value of animals to farmers. And I think because farmers see the value in the animals, then of course they would want to protect the animals. I mean, we know that farmers actually vaccinate against common diseases in East Africa and other places in Africa. There's a whole sort of really well-developed veterinary vaccine industry in Africa, and that's been going on for many years. It only happens because farmers actually buy the vaccines and demand the vaccines. But I think, like with any other intervention or implementation program, whether it's a vaccine, whether it's a food, whether it's drugs, I mean, I think you need to have very good engagement with the end users. And I mean, it's the same. It applies to veterinary species as well as it does humans. But I think going forward, because most of the technologies we're dealing with now are classed as genetically modified organisms, so there is a need.
I'm afraid those of you listening, I'm afraid George seems to have frozen on the connection. Your greatest nightmare as a introducer and questioner, especially as I can't answer the questions that have come in as well as George could. If you could just bear with us for a minute, we will try and get the connection back. George, are you back?
I'm back.
Brilliant. Gosh, I can't tell you how frightened I was that I'd have to try and answer all the questions.
Great. Did you hear all of that?
I think we did, yes. But I wanted to follow up with one question on the human side. And, we have, obviously, there's the push from public health. Human side is so much better funded than the animal side, tragically. But we also have agencies, Global Fund, Gavi, etc., which are procurement agencies, UNICEF of this world, so many fantastic agencies. Does that exist on the veterinary side? Is there a demand? Is there a pull mechanism for vaccines such as this?
I think there is, but I think I'd like to sort of think of a sort of paradigm shift in that we have been very siloed up to this point in our thinking about controlling this sort of emerging infectious diseases. So the bulk of this comes from the veterinary field. I think the organizations that stockpile vaccine for specific diseases, something like East Coast fever. But I think for this particular class of diseases, if I can call them that, there is a need for multiple, Pan-African, global organizations to actually come up with a strategy that involves One Health. Because if I can use an example of COVID, for instance, while coronaviruses, that they've caused several outbreaks, epidemics in humans in the past. I mean, the first coronavirus ever to be identified was a chicken coronavirus, and I think every species, every veterinary species I can think of has its own coronavirus. And there were coronavirus vaccines that were licensed even before we had this pandemic. But that sort of interaction between the medics and the vets, if I can call them that, is just not there. There just needs to be a lot more integration in thinking about controlling these diseases. For RVF, for instance, you'd need to think about the deployment of the vaccine for livestock and humans. And when you're thinking about a strategy to roll this out, I think you need both public health and veterinary public health personnel sit in the same room and sort of thrash out a plan that effectively sort of minimizes the risk through vaccination of, all the affected species, including humans. So I think I'd like to see something a bit like what UNICEF does in stockpiling epidemic vaccines, but incorporating more veterinary, having more linkages with veterinary, sort of actively, so that when you have a response, then you can, in some scenarios, maybe deploy the same vaccine, or at least use learnings from the control of epidemics in humans to inform the response in livestock and vice versa.
Yeah. I mean, very few better people in the world, George, for you to make that very eloquent case for the breaking down of those silos, given that the vaccine you work on is one of the not many vaccines that are actually targeted at both human and animal population, as well as all the other work you do. George, I want to move now to questions that are coming to the audience, and it's very difficult to come up with all of the questions because there are so many. But if we could go through these sort of quick fire, I will to try and answer as many as we can, and they're still coming in. So quick fire round. The first question is eradication, smallpox or rinderpest. Are there any lessons to learn from those to apply here?
I think you've got to have excellent surveillance systems. So the vaccine was fantastic against rinderpest, and that the success of that vaccine is informing the control of PPR, which is another sort of related virus in small ruminants. But to really get rid of those really stubborn pockets, you have to have very good surveillance. You have to have people who are really trained, and you have to have social engagement. You really have to engage the farmers. So I think those three things were the critical factors.
Brilliant. How long does the virus persist in meat, in domestic or butchered meat, or meat in marketplaces?
Yeah, I mean, it depends, because if we go back to the lab, to inactivate this virus, you need half an hour at 56 degrees to really inactivate it. So when there's been studies that have been done, I think as part of a developing of a vaccine called Clone 13, and basically at fridge temperatures around 4 degrees, that can last for, let's say, about 2 months. At room temperature, I'd say maybe just over a week. And maybe at 37, maybe 3 to 4 days. So I think it depends on the temperature. And so, of course, where I'm here, I've got 35-degree temperatures on average in England. I can see you're in a jumper. So the virus can survive longer there. So yeah, that's a critical factor, but those are just estimated days.
Really interesting question here. They're all interesting, but really interesting one here. If any idea if you've received the COVID-19 adeno vaccine, would that have any impact if you subsequently received this vaccine, the Rift Valley fever vaccine, using the same construct?
Yeah, that's an excellent question. I mean, I think so. The ChAdOx1 platform is non-replicating, meaning it doesn't replicate. So it gets infects in the cell. The information is delivered to the cell, and the host cell actually makes the protein, the antigen of choice. And here, we're talking about RVF glycoproteins. So, and that's what stimulates an immune response. There is very little sort of immune response against the vector because it's not replicating. So you get some limited stimulation of the response to the ChAdOx1 vector, but it's not enough to sort of affect the performance of the vaccine. And as we all know, the deployed strategy for the Oxford AstraZeneca vaccine is 2 doses, 2 to 3 months apart, and it works perfectly. So we see this as not a problem. I mean, it's been used effectively globally. So I think that sort of is a good sort of evidence that you can use the vector. Again, you can reuse the vector. Okay.
Question here from, not everyone puts their name, but Sarah Cleveland House. It's great to see Sarah listening to this talk, George. Very many discrepancies in investment in relation to vaccines administered for people or for animals. And Sarah wonders whether you had any thoughts on this in relation to Rift Valley.
Yeah, I mean, I think that's the issue has always been prioritizing epidemic diseases. I mean, I think disease X was some obscure thing that people were talking about before the pandemic, and suddenly we're dealing with disease X. I think it's the will, the political will, the sort of the public is behind this now. So we just need to make hay while the sun shines, as they say. And so I think it's all about business case, and I think there's going to be more investment, I believe, on the veterinary side of things, because this is, for a disease like this, you're really going to have a huge impact if you can include veterinary vaccination in your strategy. Yeah, because that's one, the biggest risk factor for exposure to the virus.
Question here, I hope I get the pronunciation right, and apologies if not, but from Kudos Mijigabi, apologies if I've got that wrong. Professor Warimwe, great congratulations. Your analysis of the Rift Valley treatment vaccination has been fantastic. Where do you see the best efforts for vaccine development whilst understanding the push-pull effect of zoonoses?
That's a really tough one. It's a bit related to Sarah Cleveland's question, and it's also something that I would put to, of course, Jeremy Farrar, who has an institution that would be sort of helping this pull-push debate. I think making the case for investment in these diseases in the past has been difficult, because they're restricted diseases in terms of their geography. And unfortunately, the income status of the country or the region is such that there are so many other priorities, and let's be honest, if it were to spread to Europe, to the US, whatever, then suddenly it might be right at the top. So that's the unfortunate thing. But I think the things are changing, and Rift Valley fever, Lassa fever, I think Ebola and disease X has been prioritized by the African Union, which already, this disease is being prioritized by, that sort of grouping, a global, a group that has such a Pan-African mandate and a global mandate, already influences, priorities for funding. I think it really supports, because it shows that this is a really important area for investment, and that kind of thing, I think, will change over time. But I think I'd say the future is brighter than it's been in terms of funding for these diseases. I've certainly seen that in terms of the funding situation whilst I've been in this field.
With the coming of the Africa CDC, Center for Disease Control in Addis Ababa, is there an opportunity to push that in a direction which does embrace the One Health agenda better than potentially the US CDC's done, or European CDCs, which largely are human focused?
Yeah, I think it's a fantastic opportunity, actually, and so one of the questions that we've been thinking about is so the vaccine seems to be performing very well in livestock, and is looking very good in humans, at least the early of evaluation. And looking forward in terms of how this product might be deployed and what would be the regulatory pathway. And our approach has been to involve national regulatory authorities in Africa, not be prescriptive as that this is the process that we want to use, but rather sit in a room that involves the human regulators and veterinary regulators, and just think of this as you're trying to solve a problem. You're trying to solve Rift Valley fever. You have a tool here. How best can we use this tool? And I think it will work. I'm very confident it will work, because at least then you get the views of the actual approvers of this sort of information, the people who would be reviewing this sort of information to make decisions about the use of a product, this sort of product in this setting. And I think that then feeding into the sort of disease control strategy and vaccination, because I think we are in a situation where, like RVF, there are many other diseases that would have a very poor business case for investment, elsewhere. And we need to think about the regulatory pathway for these diseases. Do we have to wait for EMA or FDA to make a decision about an RVF vaccine? So that's the sort of where we think the debate should go. And from that, I think will emerge a solid plan to take this forward. At least that's my hope.
That would be fantastic. We've only got 2 or 3 minutes left, but some fantastic questions come in from a Jeffrey Greenspan. Thank you very much indeed. Just pull out one. Does the RVF virus have one or multiple host receptor proteins, and are neutralizing antibodies directed against the receptor binding sites?
Yeah, I mean, I think the neutralizing antibodies that target the GN protein and a quaternary epitopes between the GN and GC. So the GC is the fusion protein, and the GN has a receptor binding domain. So I think that the quick answer is that you can have both, but with the GC is a bit more complicated in terms of it's a sort of quaternary structure where the neutralizing antibodies bind. The host receptors, I mean, there's been dicysine, I think heparan sulfate, so that's still not fully characterized, and I think there's still lots of unknowns about RVF virus, but I think this knowledge will improve as more and more studies on RVF. I mean, it's certainly been at the top of diseases that have been studied in terms of outbreak diseases. We're seeing a lot more outbreaks, much more at shorter intervals than they used to occur. So I think it's just a matter of time.
George, we could go on all evening. There's still lots of questions coming in, but I'm afraid we've come to the hour mark. That was one of the most inspiring talks I've heard in a very, very long time, George. Thank you very much, not only for the talk, but all the work you've done scientifically, of course, but also, and it hasn't come out in your talk at all because of your modesty, but the amount of mentoring and support you give to your students, your postdoctoral colleagues, your peers is legendary in the region and across the continent, George. And so many people have benefited from your inspiration and the inspiration of your career. So on behalf of everybody, we can't clap, I'm afraid, in the virtual world, but I'd just like to thank you, for your career, but also for an absolutely fantastic talk. And as the Royal Society 2021 Africa Prize recipient, it couldn't be in better hands, George. So many, many, many congratulations.
Thank you much, Jeremy. Thank you very much, Jeremy, and thank you all for your attention.
So, George, I'm going to also just pass on now to remind everybody, if you don't mind, that on the 6th of January 2022, we're almost at the end of the year, we have another prize lecture, the Royal Society Rising Star Africa Prize Lecture, which will be given by Dr. Naushin Gunu on the 6th of January 2022, with a title which is enticing: Nano-Engineered Seaweed for Scarless Skin Regeneration. What a fantastic title that is as well. So, George, you'll be followed by Dr. Gununu, and look forward to that as well. And almost exactly at past the hour, George, thank you again very, very much indeed. And many congratulations on this award. And thank you so much for the work you've done in the inspirational lecture. Good night, goodbye, good afternoon, wherever you are. Thank you very much. And I'll give the last word to George.
Thanks. I mean, this is fantastic. Royal Society recognizing the talent in Africa. There's a lot of talent in Africa. You're just scratching the surface, if at all you consider me talented. And thank you for the very many kind words, Jeremy. There's many of us in Africa. So yeah, keep looking on this end. Thank you.
Thank you, George.